Good morning, everybody. My name is Helen. I very recently joined the group as a PhD student, meaning 2 weeks ago. So I'm very happy to be involved. I did an internship in this group. This is why I have sort of a mini project that I can present to you. But Pavel will take over after my introduction. As you can see from the title, I will try to focus in my PhD on protein isoforms. So why do we care about this? So as you all know, the human genome has roughly 20,000 protein coding genes. However, the proteoforms, the proteome and proteoforms are much more diverse. And there are roughly 2 categories of things that contribute to this diversity. The first thing, as you have here on the mRNA level, is alternative splicing. This is a way of making 2 protein isoforms from 1 mRNA. And the second thing that can happen is site-specific features. So you can have coded in a small nucleotide polymorphism, which of course is encoded on the gene level, and then a multitude of post-translational modifications. And these things occur combinatorial, and together they diversify the proteome and make up to 1 million proteoforms. You've heard in the last couple of days a lot about PTMs and how PTMs are dealt with in shotgun proteomics. So for this part of the presentation, we will focus on this protein isoforms. So splicing is a thing for eukaryotes. In general, pre-mRNA is processed by splicing out introns and only keeping exons. However, things can alternatively be spliced, which is a way, as I mentioned already, to make proteins, multiple proteins from 1 mRNA. The most common one is exon skipping. However, there are also many different forms that have been recorded, everything from alternative 5 or 3' splice sites to intron retention or multiple mutually exclusive exons. Overall, it's been known by now that alternative splicing is the rule rather than the exception. Depending where you look in the literature, up to 95% of multi-exonic genes are alternatively spliced. So this is the vast majority, and this is why this is extremely interesting. And to make this a little bit more difficult on our side is that these splice isoforms, it's rarely that one is very present and the other one is not expressed at all. They usually exist within a ratio of each other. So can we study proteoforms of shotgun proteomics? I mean, you've seen this with PTMs. Why this is even in theory possible, it's a high throughput technique. And you can detect a wide range of proteoforms, so everything from PTMs, and now I will tell you a little bit of isoforms in an unbiased way. However, with isoforms, the same as with PTMs, the detection limit can be an issue. So usually splice isoforms are grouped together in the protein groups. So that's why you need very, very deep sequencing to actually differentiate between isoforms. So to start with my project and a little bit of what I did was first to identify which exons are present or absent within a sample. And from there on, we can make some educated guesses which splice isoforms are present or not. And for that, I used the peptide TXT file from the MaxQuant and mapped it back to the genome location to extract exactly which exon is present. And why is that even a computational thing, or why this is even a project? So there are some challenges with this. It's especially because it goes against the genetic flow. So mapping a peptide back means that you have to deal with the redundancy of the genetic code, so you can't just translate it back. It's, of course, due to splicing discontinuous, and genes are encoded in forward and reverse direction. So for my workflow, I take this peptide txt file, which is from the maxquant output file. And the first thing I do is ID mapping. So I convert the uniprot identifier to an ensemble identifier. This is more for convenience because the genome is annotated in ensemble and, of course, not in uniprot identifiers. And this is also the reason because uniprot identifiers and ensemble identifiers are connected by the gene names. I once again have to map the peptides to the protein sequence to know exactly where this peptide is in an ensemble FASTA prior. And then I also take an ensemble GFF3 file. A GFF3 file is a file for genomic locations and make a coding sequence map, and that helps me to map the genomic positions. And I also have a GFF3 output file, which can be used for visualization so far. So I will briefly go over the central part of the workflow and how a coding sequence map works. So here you have a peptide sequence of a very short one of 2 amino acids, and we know the protein which it belongs to, which here only has 5 amino acids. And then you have a coding sequence map, which looks precisely like this. It's an array 3 times the size of the protein sequence filled with numbers. And each number in this array represents the exact number of a nucleotide within the genome. So, for example, amino acid 1 here is encoded by 3 nucleotides at position 11, 12, and 13 within the genome. And amino acid 2 here is encoded by 3 nucleotides at position 14, 15, and 34. And as you can see precisely here, you would have a splice site, but we can capture it with this array. So if you have this all together, it's very straightforward. I use an exact string match to match the peptide to the protein sequence. And with the knowledge that 1 amino acid is 3 nucleotides, I can multiply the position by 3. So this is position 1 times 3 means it starts at position 3. And if it ends at position 3, I can also multiply by 3. And that's how I can extract exactly which nucleotides encode this peptide within the genome. And you can figure out if it's continuous, so if there is one splicing site, no splicing site, if it's forward, if it's reverse, all that is captured here. So I have a GFF3 output file, which can be used for visualization. I use the integrative genome viewer. I feel like if you've done transcriptomics or genomics, you might have come across this one. If you upload your file, this is precisely how it looks like. So up here, you have all the chromosomes from and down here, every little mark is supposedly a gene. And up here, you have my input file. And like this, it's not very useful, but you can zoom in. And then you get a view like this, where you can precisely see each peptide and where they're mapped and which exon they belong to. So yeah, that's how far this project is currently going. And of course, we would like to so for now, the idea is to include this workflow within Perseus, but hopefully in the more distant future, we also want to establish some metrics for isoform detection. And of course, the most interesting part is how do they change within different samples. And with that, that's my mini part. Thank you so much for your attention.
Okay, great. So that's going to be continuation of Helen talk. So basically, I just described our journey And this was a multi-year journey, multi-continent journey during my PhD, and the later revision was during my postdoctoral research. So that's deep human proteome sequencing for global detection of mutation and alternative splicing. Although, as you see, we did also detected mutations, but today I will mostly focus on alternative splicing discussion. So what's the general idea of the project? So as Helen said, in order to study splicing, you do need coverage. But in this project, we rather mean coverage, amino acid coverage. So out of 100% of amino acids which recorded in the FASTA file, how many actually covered with at least one peptide? This kind of transcriptomics way of thinking about the world. So if you take this example of the protein and digest it with trypsin, you're gonna get many different peptides, but not all of them are suitable for to be detected, right? And of course, like this is a very ultimate case. It's a very artificial case. You also have miscleavages, but let's say just these two peptides we're able to detect. So right, so now we have coverage, and by eyes you can estimate that's approximately 20–30%, which is fine, but fine for probably a relative expression analysis, but not fine for this detecting splicing, right? So what solution can be here? You can take another protease. So let's take GluC. Again, we have some set of the detectable peptides, and just a few of them actually we detect. And our big hope that's if we combine all these peptides from different proteases, we're going to get a significant boost in terms of amino acids coverage. And yeah, as you will see, that's indeed true. And if you will ask me, yeah, okay, later. So basically the design of the study is instead of just taking one cell line, one proteome, we took like 6 cell lines, 6 different proteomes. And as you may see, there's 6 cell lines, human cell lines. And this is exactly the cell lines which have been used in ENCODE project. That means that we do have super high coverage transcriptomic data. Great, so proteomes have been extracted from each of the cell line, and each of the proteome have been digested with one of the 6 of proteases separately, and each of the lysate have been fractionated up to 30 fractions, and each fraction have been measured with 3 different fragmentation methods. Fast loop forward, if you ask me if you have a limited amount of time, limited machine time, what would be the best way to get coverage, high coverage as quick as possible? I would first of all suggest to use multiple proteases. Second, fractionate them, maybe not up to 30 fractions. That sounds a bit crazy, but up to, let's say, 8 fractions. This actually works really well. And to be honest, fragmentation, HCD is the winner, and it's the fastest and the most straightforward fragmentation method. So we measured everything on a tribary system. So the second MS was measured with ion trap, and the whole data have been analyzed with MaxQuant. But just to kind of, it's a huge data set. If you take 6 cell line multiplied by 6 proteases, each protease 6 multiplied by 6 multiplied by 30 multiplied by 3, that's going to be huge data set, right? And indeed like that. So it's if you multiply all of them, it's going to be approximately 2,500 files. This is 5 terabyte data, and this is a half of the year machine measurement. So and that's it was a challenge in the first place, but be able to analyze this data, and it's actually it feels great. So, and one of the reviewer said, like, yeah, indeed, this paper described technical tour de force that generated a useful resource. And I can actually confirm after approximately published this in beginning of summer 2023. And now there's already several projects in parallel when people trying to take this data and analyze in different ways for different questions. For example, one of really like people try to detect upstream open reading frames or unusual open reading frames in the human genome. And that's data which has a huge coverage also potentially can help to find this open reading frames. But anyway, so, and I just give you an example. Of course, this is very cherry picking example. This is hemoglobin subunit gamma 1. Obviously, it's highly expressed gene. And if you take all these amino acids and color code them with depending how many unique peptides you have on top of these amino acids, you see that there's locations in the protein which covered with more than 9 unique peptides from all proteases. And obviously, there's some which have a low coverage, 1 to 3 amino acids. So it's actually, to be honest, it's actually also a very interesting task to predict which places of proteome is more detectable rather than not. So that's a separate task. But so what we did with this data, so first of all, made some observations. So if you analyze all this data separately for each protease, you see that indeed trypsin is the winner. It generates super suitable for proteomics peptides, were well detected. And but yeah, so all the rest, probably except the chymotrypsin, performing really well. And if you combine all of them, you see some advantage of combining, right? That's not super obvious here, but if you go, for example, plot like that, you take trypsin coverage, amino acid coverage, using just trypsin of all 18,000 proteins, and sort them. So from the super low coverage to the super high coverage at the end. So you'll find out that's, yeah, we're forming some well-known shape. But now, if you take each of this protein and take the rest of the proteases into account, you'll see that each dot going to be elevated on top. And that's the difference is approximately 20–30%. So we're indeed getting a boost of the coverage, amino acid coverage, thanks to the multiple proteases. But that's not everything like super sweet. So there's some problems. But that's problems which are rather not new for the proteomic field. And one of the well-known problems is transmembrane domains, which are notoriously difficult to sequence. I'm not going to propose you a solution here. Unfortunately, we're also having this problem. But in future, we're trying to deal with that. So, but let's be kind of, let's see how the large-scale problem it is. So if you take different proteases and plot the distribution of coverage for all 80,000 genes, let's say, and but you plot separately genes which have transmembrane domain, which don't have transmembrane domain, you'll see no matter which protease you're going to take, all proteases together, you see that transmembrane domain containing proteins have a lower coverage in terms of amino acids. And you may say, like, yeah, but maybe these transmembrane domains containing proteins just expressed in a low range, in a low expression range. That's why you see lower coverage. We know coverage, it depends. Yeah, go ahead, sorry. Yeah, so how the sample processing contributes to the to this bias, right? Yeah, indeed. So even though we did like in Joshua lab, it was a very standard protocol, but indeed like that was the first thing Promiga highlighted, that maybe you should play around with protocol, and that's actually what we did. But again, this is pros and cons. You don't want to increase the amount of time of measurement, but you want to get as much as you can. So, but one thing, coming back to this question, so we're not actually losing these proteins. We're not low expressed. We're normally distributed as the rest of the proteins. The issue which we have, this is actually transmembrane domains. It's themselves. So if you look at the, if you look at the peptide coverage, depending from which protease you're going to use around of the transmembrane domain, so hydrophobic region, you'll see that once you pass this border of the transmembrane domain, you see like no matter which protease you're going to use, you see low coverage. Maybe except one example of chymotrypsin, but probably you know chymotrypsin really love to digest after hydrophobic amino acids. And after you leaving the transmembrane domain, you see that the coverage is recovering. So it's just proof from my point of view that the issue is not losing these proteins, maybe partially, not that's why low expressed, but that's specifically transmembrane domains set of the super hydrophobic amino acids. It's just hard to study with current setup of the shotgun proteomics. There's some ideas how to improve it, but let's discuss it later. So yeah, so having this data set in hand, so we decided like, okay, what should we do? We have a hammer, give us the nails. So, one thing we decided to do, we decided to jump into the alternative splicing, which I feel like from proteomics point of view, it's heavily underdeveloped field. Of course, people in transcriptomic field do see a lot alternative splicing happening, but for them, it's a big question, is it kind of somehow translates to the proteomic level, right? People did actually many cool studies with ribosome profiling, where we were able to see if like ribosome profiling scanning these alternative splicings. But at the end of the day, we all would like to ask, is this kind of all this variability translated to the protein level? And having this data set with 80% median coverage for the all 18,000 genes, we decided like to look at this question. Okay, great. So, one thing I have to say in advance. So if the trypsin is not the best choice for the study splicing, it's a bit controversial, but there's actually one, let's say, funny and sad reason for that. So, but if you try to map, as Helen described, if you try to map your peptides to the genomic coordinates and you focus on distance between 5' of the exon to the N-terminus of peptide in terms of genomics coordinates, you will find out that there's a really high peaks when you have a distance of 0. So what does it mean? That you map your peptide and N-terminus co-occur with 5 terminus of exon. It's actually also true for the C-terminus and 3'. So you see that the distance is 0. What does it mean? It means that we do have many non-informative peptides which do not support one alternative splicing over another alternative splicing. And there's a definitely bias of, we don't have so many splice peptides. We're underrepresented. Let's put it this way. But why it's happening? That's another question. And why it's happening, it's actually really interesting. It's, you know that the splice machine recognizes specific nucleotides in the intron region, at the beginning of intron region, at the end of the intron region. But the truth is that there's also a bit information, a bit of a small bias of nucleotide presence on end of the exon and beginning of the exon. And this small bias making some amino acids occur more frequently, and you know the end story is arginine and lysine really love to be there. So basically, if you think about this, between exons, frequently there's arginine and lysine happens there. It's a really very unfortunate bias for us, but that's happening. But that's yet another proof that's saying us that if you want to go and study splicing, and especially alternative splicing, you need to go into multi-protease approach. So yeah. Okay, but let's go through the example, like what does it mean to detect actual alternative splicing? So imagine that this is amyloid precursor protein. It has like multiple exons, but let's focus on exon 7, 8, 9. The transcription has happened, so premature RNA is here against 7, 8, 9. But during maturation, this RNA can end up either skipping exon, so having 7 and 9, or no skipping, so basically merging all these exons together, so 7, 8, 9. And what we kind of would like to see, we would like to see proof on a peptide level for one alternative or another alternative, or both of them present. So that's what's actually we are looking for. So we are looking at the specific peptides which cover this scar between exons, so this exon jump. So the peptides which are equally present in exon 7 as well as 9, so which support exon skipping, or 7, 8, as you see these peptides, and 8, 9 as these peptides. And of course, for all these peptides, we do have a spectra, which is a high quality. And but I would just like to highlight that we, at a certain moment of the project, decided to make one definition separation. So let's say we're fully detecting alternative splicing. We are fully detecting splicing only if we're detecting peptides which support exon skipping and no skipping. We are partially detecting splicing, alternative splicing, only if we're detecting either exon skipping, either no skipping. So it's actually important difference because like fully detected alternative splicing, it's where we actually saw both alternatives being present, either in one cell line, either in several cell lines. Okay, great. So as Helen discussed, like there's a really indeed dozens of different alternative splicings happening. So we detected many of them on different levels, both detected, partially detected. We compared the transcriptomic data, but today I would like to mostly focus on skipped exons. So it's basically, it's another name in Helen presentation was cassette exon, but as usual in this field, there's multiple terms for that, but let's call it skipped exons. So one thing we kind of found out, thanks to our collaborators, that's there's one particular type of splicing, which is apparently frequently happening. It's called out-of-frame alternative splicing. And we proved that this out-of-frame alternative splicing is not present on a proton level. But okay, let's start simple, right? What's this out-of-frame splicing is? So if you look at the in-frame splicing, so it's a splicing which exon exclusion splicing, which has a middle of exon, the length of which is dividable by 3. So it's the length of this is either 6, 9, 12, 15, etc. So it's, let's say it's a good alternative splicing. Why do I say this? Because the frame shift is not introduced when like ribosome going through. So it means that in the case of exclusion of exon is going to be AS amino acid sequence, but during inclusion it's going to be ASMS. So now you know on which conference I presented this slide, but but you see like this middle exon have to be divisible by 3. The length of that have to be divided by 3. So, but alternative is out of frame. What is happening? So although sometimes ribosome can go and sequence codon after codon on inclusion of the exon case, you see that when we merge on a last exon, that's going to be some difference, and this difference is frame shift. And usually, one of this alternative not going to be stable. Usually, introduction of the frame shift destroys protein, and that's what actually we see. So we see that in the case of in-frame translation, in-frame alternative splicing, we do observe dozens, like 22% of all in-frame translations who do have a support for both alternatives. We're fully detected. So we do observe both alternatives. But in the case of out-of-frame translation, we do not, we almost never detect both alternatives. So from our point of view, that supports the hypothesis in the field that out of two alternatives, one alternative constantly degraded. So another question is to ask why cell is doing that, but that's actually a difficult question. So, but there's some studies on top of that. So, but this is an active regulation of protein synthesis, but using alternative splicing. So, but at the end of the day, we decided to kind of find out which factors impacting our capability to detect both alternative splicing using proteomics versus non-detected alternative splicing. So what I mean by that, we just basically made a binary classification using XGBoost, which tried to separate using different properties of the alternative splicing, tried to separate alternative splicing which we are fully detecting and partially detecting. So what kind of, and if you ask like functionally, we're asking which kind of properties of alternative splicing define, like increasing our chances to detect them on a proteome level, right? So usually people doing this manually by hands, but we decided just to ask machine learning to do that. So, but obvious features which are properties which are happens to be very important. This is transcription abundance. So means that more transcripts you have, higher chance that we will detect this alternative splicing. There's another definition called percent spliced in, PSI. I will discuss this later, but this is yet another side of the same coin as a transcription abundance. And the frame status. The frame status we discussed with you, so it's being either in frame and out of frame, and as you see, this has a huge impact. Will it be detected or not? Okay, great. If we focus on transcription abundance as a factor which defines will we detect this alternative splicing or not, we obviously see that higher transcript is abundant, like more and more transcript is abundant, a higher chance that we detect both alternative splicing on proteomic level. So there's no surprise here, right? Proteomics, as any another omics, we do have bias detects something which is high expressed, and alternative splicing is no exception. Go ahead. Yeah, so okay, you remember this story, right? APP. So I'm saying that if fully detect alternative splicing is the one where we're detecting both alternatives. A partial one is the rest. So the partial is usually the ones we're detecting at least one, just one alternative. So we just would like to separate, we would like to have a really strong and solid cutoff between canonical splicing and alternative splicing. And we are doing so introducing these two definitions, basically. Yeah. And Okay, and the second thing is a percent spliced in. So I hope you don't mind if I draw on the board. So I will shout a bit. So, but imagine we have a splicing. We have 3 exon situations, and I call them exon A, B, C. So what we can do as a cell, we can exclude exon C. And another option, we can include exon B. So sorry, we can exclude exon B, sorry, or we can include exon B. So, and what's a percent spliced in means? So that's, it basically says what's the number of ABCs. So how many, in case of transcriptomics, is how many fragments support inclusion of the exon B versus 100%. So basically versus ABC plus AC, sorry. So let's say, like from proteomics point of view, which alternative is easier to detect? I would argue it's easier to detect alternative A, B, C, so inclusion of the exon. Why? Because you have many possibilities to do so. You can detect either jump from A to B, or internal or fragments which map uniquely to the B, or B to C. But you have just one chance to detect exclusion of this exon. You have to detect peptides which are jumping from A directly to the C, right? So that's why what we found out, that's what we frequently observed, that we indeed have a bias towards detecting inclusion rather than exclusion. And this plot actually tells us that if you want to equalize chances of detecting one versus another, you rather have to have expression, you have to have expression of AC twice higher compared to the ABC. It's slightly counterintuitive, but that's basically equalized chances from proteomic standpoint of view to detect these splicings, so these alternatives. But from another point of view, let's say one thing. Imagine why detecting splicing is exceptionally difficult task. We already see that we have a bias towards highly abundant genes. Okay, but there's another issue. With alternative splicing, you're basically diluting 100% of expression of gene towards some alternatives, right? Imagine if you have 10% and 90%. So cells decide to include in 10% of cases and 90% exclude. So what if you want to detect fully alternative splicing, you need to focus on this 10%, which are low abundant. But from another perspective, we made our task way more difficult because we diluted initial expression level 10 times, right, towards one alternative. So that's why actually alternative splicing is hard to detect. It's because of the bias versus against trypsin and arginine lysin, and because of the you may think about alternative splicing as a further dilution of initial expression of gene. So it's just amplify the problem. So coming back to the question, okay, what if we take trypsin, or if we'll take all the rest of the proteases, what would be the splicing coverage, alternative splicing coverage in overall? So, but the thing is that no matter which protease you're gonna take, you're gonna have a coverage in average for even highly abundant genes, approximately 20–30%. But if you combine all proteases, then it's gonna be 40%. So there's many questions here, but the main question is how can we increase from 40% to the 100%, right? It's yet open question and yet to be solved. But what I can tell you, so there's a paper out, so if you want to read this, so you're more than welcome. And yet another thing I would like to highlight, that yes, yeah, that was a bit sad story, but we found out we basically we made embroideries with my wife. I was making like a surrounding red line. She was actually making the stitches inside. But you see, it's actually super cool piece of art we came with. But unfortunately, editors of the journal said no. But you see, like, if there's so many details I love. So first of all, you see inlet of the mass spectrometer. You may recognize this light of the HPLC. So you see there's a different proteases lying here. And there's even some proteases, like you see, like scissors. So there's a previous proteomes, like proteomes using just trypsin, the proteomes which using trypsin and ASPM. And you see, this is the best proteome in front of you, of course, because we stitch it for so long, we spend so much time, and you see the full completeness of the human proteome. I described really well to editor, so, and I was... Yeah, I have to highlight this was a work of many people. So, and actually multiple hours during the night and evenings. We work on that 2 weeks basically, but no is no. Yeah, the thing is that we were thinking like one of the big, I feel like, disadvantage of the previous studies of which was published, it's just lacking quantification. So there's a lot of identification things. We literally trying to improve identification, but there's no quantification. As a result, there's no way to apply this well. So, and in the version 2, we decided like to combine the power of different groups. So we decided to improve sample preparation, LC, MS instrumentation, improve or play around, let's say, put this way, and software development. And at the end, we kind of come up with the idea that we would like to study the embryo development. So basically, we know that during the embryo development, splicing is going around, especially alternative splicing. And indeed, the brain has a huge amount of splicing happening. And we believe that during the embryo development, we will be able to quantitatively detect this splicing. And who knows, we may make pull downs, alternative splicing specific pull downs, which allows us to see interactomics and depending on the alternative splicing. So, but that's project ongoing right now, and let's see how it goes. Yeah, so this work is definitely would not be possible without Jürgen and without Kuhn Lab.